Vienna at this time with all these kind of ideas, and Freud doing his thing. And, it's a really cool place, and Vienna is beautiful too. Nice time. But anyway, so Paul Broca came, and he was like one of these jack of all trades kind of scientists. Like he's the one that identified the problem with rickets. So everybody will know what rickets is. It's a bending of the bone. So people walk around with like bent legs like this, like they're riding a horse for a long time. But through his observation, he identified that as a problem of malnutrition, the lack of vitamin D. So he also was a big fan of Darwin. So he also created a lot of evolutionary type of theories. He's the first one to actually put genetics in psychology in a way. So he's the one that really pushing this idea of heredity and the passing on of traits from person to person through this idea of genetics. He also he was the one that passed the idea that a lot of cancers were driven by genetics, because his wife was, her family had a history of cancer, so it was likely that she was going to get it. So he really studied a lot on cancer and that. And it was just, with Broca and his kind of, just his curiosity, he worked in an insane asylum, and he was really noticing, a lot of these patients that would come in, and through kind of this way, like until he had this one guy, Tan, come in. And does anybody know the case of Tan? His name really wasn't Tan, but that's what he called him. It's because that's all he could say. When Broca would speak to him, he'd be like, Tan, 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 Tan. I wish I could give my lectures like that sometimes. Or maybe that's what my lectures sound like to you guys. It's me up here being like, Tan, 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 Tan. But he was really curious in this because he was expressive, like he's emotion, but like you could understand what people were saying to him, because he could do other ways of communication, obviously with his hands and with writing, but his speech production was absolutely wiped out, and that's all he could say. And then eventually, he's an older fellow, so this likely happened from a stroke, and he eventually died, and they did an autopsy on him. And then they found this area right around here was severely damaged by this stroke. So then he's just like, this is his first insight into localization of function, is this particular brain region, and he's likely seen other brain regions and brain atrophy. Nothing to do with his this language deficit, but this area that he termed Broca's aphasia and Broca's area was the first kind of pinpoint of like clinical pathological correlation method to infer specific regions of the brain that were necessary for certain higher functions. Because this is absolutely high function when you think of taking in language, doing whatever the brain does to language, but the actual motor output is that output is at the end of the hierarchy of thought. Okay. So this was really pushing forward the idea of localization of function. So I'll give you a little. Anyways, I had a little video. Oh, there's a video example. Should practice this because it can pain in the butt. Anyways, I'll move on from that. But you can see some examples. Does anybody want to read this out for me? Anybody?
Yes.
Okay.
Monday are Dad and Peter and Dad are hospital, and Wednesday 9 o'clock, and Thursday 10 o'clock. Doctors, twin doctors, and their teeth. Yeah. Smart university, smart, good.
That sounds like Trump.
Strong words.
His sense is very
good genes, very good genes. Very smart. We're in School of Finance. Very good, very smart.
Wait, did you put that on purpose?
Yeah.
Oh, I just
that is Trump.
Yes, that I got.
Yeah.
But I didn't know if it was actually expressive aphasia. Never mind.
Yeah. So there's one example here of actual expressive aphasia, and these two ones here are Trump. Actually, in a recording. I feel bad. I hope there's, Trump supporters. I never liked Trump before the presidency. So it's nothing to do with because he's president, it's just, he's always annoyed me. Either when he was in wrestling or when he was on, you know, his television show. Anyway. So with Broca, he really started, you know, creating more boxes around stimulus kind of output. He never really tried to look at, you know, making connections or anything like that. He just identified there's certain these faculties in the brain that are responsible for these particular functions. And in terms of language, we know the different functions of language, such as articulating, the motor output, and also, see, look at my language, it's failing on me right now, but, you know, interpreting language and that sort of thing. But the one problem is, you know, a lot of people today criticize this sort of modeling approach is, you know, where is Broca's area? So there's a lot of different definitions, and this is a problem with neuroimaging of today, is to try to identify what brain region. So this really nice review paper kind of went out there, and any paper that, you know, was saying they're looking at Broca's area, they looked at their R01s, or they looked at the atlas that they used to define it. And so 50%, you know, would focus on this region, but you can see the diversity across papers using different regions, all kind of around there. But when you look at the two most, you know, this could be an area, but, you know, this whole area here cannot be contributing to our motor output of speech. You know, there's some likely areas in there that is highly relevant for that, but, you know, it's just really hard to pinpoint it, okay?
Yeah, was this like a meta-analysis in the review?
Yeah.
Okay.
Yeah, I could send around the paper if anybody's interested in
So
it.
this would be a case of convergence.
Yes. Okay. So following Broca, you know, again, you know, Broca was highly regarded in his kind of his technique and really influenced a lot of people. And one of them was Karl Wernicke. Okay. So Karl Wernicke, you know, is again, you know, working in these insane asylums, seeing a lot of these clinical patients, and, you know, found that, you know, this aphasia is not just motor output, but it also could be the input as well. Okay. So same sort of measure, you know, he'd identify these kind of aphasia people, and then some of them would die, and then he'd do his dissections and see that it's likely this area of the brain, particularly the left side, was really contributing to the aphasia. Okay, so it's like the input from the sensory systems to this region that would, you know, allow us to recognize verbal speech. Okay. Maita, do you want to take this one away?
Sure.
So look at the difference between Broca's and Wernicke's aphasia.
Broca's aphasia. Cookie jar, fall over, chair, water.
Okay, so before I interrupt, so the purpose here is for the subject to explain what's going on in this picture. Okay, so that's, sorry. And Wernicke's aphasia?
Well, this is, mother is away here working her workout here to get her better. But when she's looking, the two boys look in the other part. When they're small, tile into their time here. She's working another time because she's getting to. So two boys work together, and one is sneaking around here, making his work and his further fanas, this time he had.
Where's the cookie jar? And where's the second boy?
Well, maybe,
Oh, the cookie jar is in the cabinet, like that's
Yeah.
what they're reaching.
That's what he's
for.
reaching
sorry, I
for
couldn't
there.
quite see it.
yeah,
Okay.
sorry, it's
And I
not
think
the
it's a
greatest
vinegar.
picture.
Okay, now I see it. Okay.
And then the mother is just probably on her high on Valium pills and not really noticing, you know, the water spilling over. But you can see, like, with Wernicke's aphasia, it's kind of fluent. He could, you know, produce language, but it's not very coherent. Okay, and this is something to do with, you know, Wernicke's area, obviously, and Broca's aphasia. It's just not making any sense at all. Like, he can recognize what's out there. He's kind of understanding what's kind of going on, but he can't, you know, produce the motor movements to really, you know, produce, you know, a coherent story here. Okay?
Do Wernicke's aphasia patients understand things properly?
Yeah.
easily? Okay.
But it's in terms of trying to generate exactly what they want to say.
Yeah.
And they could send it to the motor output, but what they're sending to the motor output is impaired.
Has anyone taught them like ASL or something after they have this aphasia?
ASL?
Yeah, like the sign language or something. Can they develop new language in a different, like use a different motor?
That's a good point, but at these kind of levels, right?
Yeah.
What the problem is, okay, well, maybe with Broca's aphasia, but possibly not Wernicke's aphasia, because their sign language would also be broken. Because what you want to conceptualize and think is fine, like, you know what you want to say, but then when you send that, it's just all messed up. So in sign language, it would be the same sort of thing. You're sending the output of language, but that output had received wrong information. But the idea, the key things here again is these particular functions that are disrupted in these clinical populations have precise or somewhat precise lesions. Lesions are never precise. You just don't go in there with some sort of laser pen and cut out a brain. Like when you get these strokes and stuff like that, you know, you get a lot of regions infected, but you know, likely a great deal of Wernicke's or Broca's is impacted, especially because those regions are close to, you know, big vascular systems. Like not all parts of the brain have these big, you know, highways of blood running through them, but there's particular parts of the brain that are more vulnerable for these sort of strokes and have more blood pumping out. And one person told me what a stroke is like is if you have a hose and you start having a hose and spraying a sand pile. You know, that's what blood does to the brain. Just kind of, you know, wipes it out and kills it off. You know, gruesome. You know, I don't want a stroke. I'd rather have a heart attack. Anyways, so here's another example here. I think these are kind of fun to kind of understand, you know, what we take for granted every day, what we're slowly losing possibly. You have a very good aphasia voice.
Thank you. Thank
You're
you.
welcome.
What kind of work did you do before you came to the hospital? Never now, Mr. Oye, I want to tell you this happened when he rent. His cal come down here and he got rent something. It happened in this rock here's where with him or his friend like was. And it just happened, so I don't know. He did not bring around anything and he did not pay it. And he run all of these arrangement from the Petus on from his Pessit in these floors now, and so he had them around here.
But can you imagine how frustrating it would be for this individual? Like, to him, maybe he's making perfect, clear sense, but to the person he's speaking to, just word salad, you know?
Isn't this how all grad students feel when they talk to PIs? Like I do want to explain something and just say it.
Yeah. Sometimes it's the other way around as well. But again, from this really nice meta-analysis paper, you could also see when people are trying to discuss Wernicke's area, you know, it's not a clear region, and especially with Nielsen's 1946 one. It's a very small region. But what we know about the auditory cortex and, you know, the other cortical regions is like the auditory cortex is nice and topographically mapped. You know, it's very well organized and structured. And its projections are very important. And like, who knows, you know, like back in this day, what's important here is that they're creating these boxes, but they're actually tying these boxes to anatomical structures. Whether it's, you know, the first one or second one, it just doesn't matter. It's this idea source localization. But also, Leithein's model is he's creating these paths. So he's creating a nice, well-structured model, leveraging the work from the man Wernicke. And as Ian pointed out in one of his discussion classes, one of the reasons why Wernicke was so well famed is because he wrote nice. He wrote so people could actually understand what he's talking about. And that's the power of writing. You know, if you really want to have your message sent out and relayed across the scientific community, write nicely, write straight and forward. Like sometimes I've read papers, really good, awesome papers, and they're just like, oh my god, I could barely get through it, and what you really retain from it is not exactly what you should be retaining from it. But anyways, that was the power of Wernicke, and Wernicke really influenced Lifeline. Is that how you pronounce that name? It's German?
Lichtheim.
Lichtheim. Yeah. And so he's the one that really developed Wernicke's model into what it is today, and what Roger Graves' paper really talked about, creating these pathways, understanding what areas correspond to what areas in the model, and what happens when you lesion, not just the region, but its connectivity, and what kind of clinical symptoms or pathology that actually produce. So this, to me, when I was in grad school and I took Roger's class, and when taking Roger's class, he was really proud of this paper. He's an old school neuropsychologist, and first time I met him, I walked into his office and I'm like, hi Roger, nice to meet you. Is Dr. Graves to you? Okay, so that's the kind of persona he was, but he was really nice and really well knowledgeable, and really got me thinking about how to tie these cognitive models into the brain and kind of use it to really understand clinical symptoms, okay? And again, you can see like with Lifelines, like he was really all over connecting the dots, making these paths. What happens when you cut the articular fasciculus between A and M? You get conductive aphasia and these sort of symptoms. And that was his big thing, was conductive aphasia and looking at this sort of pathway here. And back in that day, that was the pathway, okay? And it just connects, Wernicke's to Broca's. But what we now know through a lot of connectivity methods is that the connectivity pattern between these regions are very complex, and they go all over the place. And they take in massive amount of information and relays it to particular brain areas to create these high-level cognitive facets. So you can see the articulofasciculus right here. But a lot of times, like, what I'm really curious about in my own research is, we always think like the brain is these compartments, right? And these boxes that are producing these models. But the brain is so massively connected. Like you get to this guy up here, it goes right from the temporal poles right into the frontal cortex. Where when you really look at the brain, like in this next picture, for example, you always think like the temporal cortex, because of this kind of structure, is kind of cut off here, right? I hope everybody doesn't think that. It's not cut off. It creates a one big circular loop. Okay, so this information could just be spinning and spinning and spinning, and there's no particular end to any of these patterns. They just kind of carry on. Even like for vision researchers, you get these mass projections right from the occipital cortex right to frontal cortex, no stop. So there's some sort of communication between these high-level prefrontal systems and very low level that doesn't have to go through a lot of these association areas, okay? And so through this work, through this model, and through this particular patch of history in our sciences, is that this is the development of cognitive models and cognitive science. And I think a lot of people suggest it's the birthplace of neuropsychology, but I think it's the birthplace of cognitive neurosciences, where we're creating these kind of models of high-level functions, really tying it down to what we know of the brain. And the more we know about the brain, the deeper that we can actually go. And then using this sort of model, if you guys, this is the answer to the last question, what happens when these regions get knocked out? Through lesions, through TMS, how does that affect, the inputs and outputs of the actual high-level cognitive function that these inputs are actually generating in the brain, okay? And from this point forward, you get, this mass production of really good scientists like Hermann von Helmholtz, You know, he had this resonance theory that the brain acts as like a tuning fork where different frequencies are passed along the brain. And at this point, he's developing things like psychophysics that we can measure these kind of these resonant theories and using really strong experimental paradigms. And this is kind of the idea of what we know of oscillations today. Because oscillations are frequencies, they're used to pass information and drive connectivity. And he used the idea like tuning fork, where his experiments were hitting a tuning fork and showing that this could resonate at 8 hertz, but it also could also resonate something at 16 hertz, for example, just because it shares similar properties. And then William Wundt, you could go and see this particular laboratory in Leipzig. That's where it was, right? And then, you know, I'm going to kind of cut it down from there because I think we're getting to the point where you should be well familiar with what's going on in the turn of the century with the big divisions between behaviorism and cognitivism, generated by people like Skinner. So if anybody so Skinner was a failed writer, and then he went into the sciences or behaviorism. But his writing, his creativity never left him. So what he took from all of his experimentations, he wrote this book called Walden 2. Has anybody read it? It's very interesting. So what he's created was like this guy from the Western world goes to this place, this utopia, that's all based around behaviorist ideas. So, you know, things like child rearing, work, gender equality. It's all, you know, based around Skinner's thoughts, how the world should be. So if you want to see what a behaviorist world is, read Walden 2. It's actually pretty interesting. But, you know, with with Skinner, he was really interested in the input and output and wasn't too interested, as you should all know, about what's actually going on in the brain. The brain wasn't really important because all you really need to do is kind of really shape behavior based off of the environment. And of course, Tolman comes around and rejected this idea and used, you know, similar experiments to do it, such as maze-type paradigms. But through this work, through the behaviorist and cognitivism, came like the maze debate, or what we generally have, like these two like very principal theories about reinforcement learning and cognitive maps, and we're still studying it today. And they're still basic on the ideas that are produced by these two fellows here, Thorndike and Tolman. But what really kind of paved the deal was this guy. Does anybody know who he is? Who?
Chomsky.
Yes, Noam Chomsky. So he's, you know, back in the 1960s, produced this argument against the behaviorists that he was successfully, and he actually won. Okay, and I can't get. So look up the debate. Okay, if you're really interested in him talking about this kind of time in his life where he was really not really, you know, shut the behaviorists up, but also telling the behaviorists like you really have to, you know, take in the brain as an important aspect of your experimental and theoretical paradigms. Okay, and then so this I just want to kind of leave it at that. I don't want to lead into Galit's time, but you know, this is what kind of cognitive neuroscience kind of evolved from, and it's still evolving today. And I didn't get into the M&I without Penfield, but I'm sure you guys heard his story, or Brendan Miltner. One thing I want to talk about is convergent and complementarity. Is, you know, this is why cognitive neuroscience is hard. These are the tools that we have to work from to try to really grasp what the input is, what's happening in the black box, and how the black box is producing its output. But to really understand, you know, any of these methodologies is quite difficult, especially for a person to really do, you know, animal work and human work is I don't know, it's possible, but it's probably best to do it through collaboration. But, you know, this is why, to me, cognitive neuroscience is not a soft science. It's a science that involves a lot of different angles, and it's hard because, you know, you look at the tools that we have to use, and You know, even though the pulse signal is slow, it can still tell us where localization is actually occurring. You know, and that's important for developing a lot of models and theories as well. EEG is the opposite, for example. We got really time, frequencies. We could understand the rhythm of the brain by just looking at EEG signals, but we don't have a lot of confidence where these signals are actually occurring. But then that's where the convergence and the complementary kind of methods come from, where we could use each of these techniques at the limitation of others. You know? All right.